Robinson here from American Sentinel Canine. So last night, Brandon Edmonds and I had a live. I thought it went pretty good, but I wanted to discuss briefly a little bit about canine genetics because at the beginning of the video, he asked me if I was a canine geneticist, and I changed the topic to say, look, my master's degree and my bachelor's degree are in animal sciences. We studied ethology, we studied genetics, we studied ethology, animal behavior, we studied reproductive physiology. These are various aspects uh, that a person who is considers himself or strives to be, I should say, an expert in genetics should get an understanding of. But I wanted to address why I did not specifically say, yes, I'm a canine geneticist, because the truth is there's levels to this. And that's something that a lot of people don't really understand. And so let me just give me a minute here as I share this topic in a few groups. I want to get this out there because it's an important topic. We're going to discuss what is a canine geneticist and how are there different levels to this topic and what the different levels are. As far as levels go, imagine the difference between a mechanic, an engineer, a guy that can change oil but he's not really a mechanic, and then an owner that might know how to put air in the tires. All right. These are not all at the same level. You know, an engineer is a person that designs an engine. In my opinion, a canine geneticist is like an engineer. They understand the subject completely from end to end. And sometimes a geneticist may not be a breeder because they may not have husbandry skills. They may not have animal handling skills, but they certainly can understand. They may not understand how to assess traits properly, but they may understand the aspect of the science behind it. But that doesn't mean that they necessarily can do it in the field. Then you have a master mechanic, a true person that they understand what the engineer created. They may not get all the science behind it, but they certainly can communicate with an engineer. They can take the engine completely apart. They can reassemble the part. They don't have to be told what the part is. If they find a new engine, if they're a master mechanic, I'm not a master mechanic by any means. Okay, I don't want to misrepresent my skill set. I am not a mechanic, but I can remember one time back when I was in college, I worked in apartment maintenance. We found this old two-stroke motorcycle leaning up behind a bush at an apartment complex that had been abandoned. And it was obvious it had been there for years and years. Vines were growing over it and whatnot. I pulled the VIN number off of it and contacted the uh, vehicle, whatever DOT, I guess it was, Department of Transportation or whatever it was, to try to find who the owner of the vehicle was. Let them know, hey, their motorcycle was here. Did they want it? In the meantime, dumped the gas out of it, tried to crank it, put new gas in it, tried to crank it. It wouldn't run, and we noticed it was hot wired. So we figured it was stolen. Well, I don't remember how exactly I was able to find out who the owner was because I do remember them telling me that they're not supposed to be able to tell me who the owner was. But regardless, I did find the owner, and I think we told them that we found it hot wired at an apartment complex, and maybe that's how they really gave us the information or whatever. So I called up the owner and said, "Hey, we found this motorcycle behind this bush at the apartment complex, and it was hot wired." And he's like, "Yeah, my motorcycle was stolen years ago." And I said, "Well, do you want it?" And he said, "No, it's this was like a 125 Suzuki, and it was probably 15 or 20 years old at the time, and it had probably been sitting there for 5 years." He's like, "No, I don't want it." And I said, "Well, I'd like to try to fix it if you don't care." And he said, "No, I'll be glad to fix it. I still have the uh, title." And I was like, "Cool." So he sent me the title, and I took it home, took it to my garage, and changed the oil. It was a two-stroke, so you didn't change the oil, but I changed whatever fluids it had, in the gearbox or whatever, I'm sure, put gas in it, put a new spark plug in it, and tried to fire it up. And I made a strange observation. All 5 gears ran in reverse. So I called up a mechanic and I said, "Hey, I got a motorcycle and all 5 gears run in reverse." And they said, "That's impossible. That cannot happen." And I was like, "Well, you can tell me it can't happen, but I'm sitting here looking at it, and that's what's going on." And they're like, "That just doesn't make sense. That's impossible." So that made me lose faith in that shop, and I thought, there's no need in me taking this motorcycle to that shop because they said it can't happen. They don't have the skill set to fix it. So I was like, well, I got it for free. I might as well just take it apart and look for something that doesn't look right. So I took it apart and I came across this piece that looked like a half a nickel, and it was sheared, broke off. I was like, I don't even know what this is called, but it's broke. So I went to that shop and took this piece in, and I said, I need one of these. I didn't know what it was called. And he said, okay. And so he said, what kind of bike is it? I told him what it was. And he went and he got the part, and he came back and he said, you're the guy that's got the motorcycle with all 5 gears running in reverse, aren't you? And I said, yeah. And he goes, I said, how'd you know? He goes, because I talked to my master mechanic back there. And he said, well, actually, all 5 gears can run in reverse if there's a couple of conditions. One, it has to be a single cylinder motor. Two, it has to be a two-stroke. Three, the timing has to be so premature that when the piston's coming up for on the compression stroke, the spark plug fires, sends the cylinder back down in reverse before it reaches the apex. And then, because a two-stroke, one-cylinder engine the engine can run in reverse if the timing is so premature that it fires and reverses a cylinder in the piston, reverses the piston in the cylinder. And I was like, interesting. So I went home, put this piece back in, reassembled it, put the timing where it was supposed to be, fired it up, and it worked. After it did that, I went to a hardware store, got a key, electric key for like a garage door opener, turn it on, turn it off, or whatever. And I got you was going to say not able to happen, but it makes sense. So we put the key in it, and I got a free motorcycle out of the deal. Now, does that make me a mechanic? No, that makes me more like lucky, I guess you could say, and observant. Um, and this is something that a scientist can kind of do, but it doesn't make me an engineer. Doesn't even make me a mechanic. I don't know what the pieces are called, and if I had too many pieces laid out on the floor, I'm sure I wouldn't be able to get it back together. But a master mechanic, like the guy he talked to in that shop, that's a master mechanic, because the guy that was on the phone was like, this doesn't make any sense. Went and talked to a master mechanic, and that guy told him how it could happen. That would have been the guy to fix the bike. So this is what I wanted to discuss briefly when it comes to canine genetics. Do I consider myself a canine geneticist? In some degrees, no, and in some degrees, yes. It depends on who you're talking to and what their level of expertise is, and it's all relative, so to speak. I wouldn't go to a person that specifically studies canine genetics and knows all the different traits that a dog has, whether they're monogenic, polygenic, dominant inheritance, multiple alleles, and things like that, gene manipulation, um, where they are able to modify the genes, knows the mode of heritability, or the heritability index for all the different various traits and whatnot. Because frankly, I'm more like a master mechanic when it comes to genetics than a geneticist, because a geneticist is going to they're going to understand all the deoxyribonucleic acid aspects, where the adenine, guanine, thymine, cytosine, and whatnot, and they're just going to take it to a different level than I would. But they won't know how to read a dog's behavior. They won't know how to do selective breeding. So one of the comments here was a guy said, this guy's a mechanic. He said, LOL, 22 years as a ACE or ACE. I don't know if it's ASE. Excuse me, ASE diesel mechanic. And then he replied next. He says, we don't like engineers. That's kind of how a professional dog breeder is with a canine geneticist. A professional dog breeder can understand the engineered aspect of genetics, but they don't necessarily understand all the things that are required to be a geneticist. But they do understand genetics and breeding. And you'll notice the avatar for this video, or thumbnail, I should say, for this video had an egg, and sperm going in there for genetic recombination, which means the sperm carrying half the DNA, the egg having half the DNA, both of these being haploid cells. And when they recombine, you create a diploid cell called the zygote. And that's the first stage of the new offspring. And, it's kind of like that. You have to understand selective breeding. So I have learned in this field of dog breeding, there's a lot of people that can't even put air in the tires that call themselves a genetics, which means all they basically can do is say the word. They can say the word genes, they can say the word genetics, they can say the word trait, they can say the word desirable trait. They might be able to say, we look for desirable traits. But the truth is, they don't understand any aspect of genetics. They don't understand the founder effect. They don't understand what outcrossing does in terms of gene flow. They don't understand what selective breeding does in terms of genetic drift. They don't understand the difference between polygenic traits and monogenic traits. The vast majority of them don't know how to do a Punnett square. I saw a guy recently on one of the dog groups saying that, excuse me, they were mocking someone that said someone was claiming that two fawn dogs cannot reproduce a black, and they were harassing that person. Well, it turns out that person's actually right 99.99% of the time. And the reason why I say that is there's two versions of black. There's what's called the dominant black and then our recessive black. But in the American Pit Bull Terrier, I don't believe the recessive black gene exists. I have not been able to verify that with proof, but I think the only black that you see in the American Pit Bull Terrier is what's called the dominant black. As a result of that man's right. Two fawn dogs cannot produce a black. However, what that person doesn't understand is they said, well, I've seen it before. Well, what they've probably seen is what's called a red nose or a chocolate. And a chocolate is actually not a fawn dog. It's actually a black modified dog. So when you have a chocolate or even a tawny, you actually have a black dog that's been modified where the black coat, which the dog has, was modified to a brown coat. That's the chocolate. And then a tawny is like you have another modifier, the blue modifier. So if you have a black dog and a blue with a blue modifier, it turns the black to blue. Well, the black can be on one. So let me show you a picture here. I'm going to do a screen share to show you what I'm talking about. We're going to go to a page on my website. We have a topic called Canine Genetics. And this picture here, we have what's called a karyotype. A karyotype is a picture of all the chromosomes. So you'll notice there's two chromosomes for chromosome number 1, two chromosomes for chromosome number 2, and we have a total of 39 chromosomes in the dog. Now, a human has 23 pairs of chromosomes, but a dog has 39. They actually have more chromosomes than a human being. They don't have more genes, but they don't have more loci. Well, let's see, they may. I think a human has 22 or 24,000, something like that, loci. I think a human has, and then the dog has either 2,000 more or 2,000 less. One of us has 20 or 22, and the other one has 22 or 24,000 loci. Now, what is a loci? A loci is an address of a trait on a chromosome or in the genome. So when you're looking at all of these chromosomes, we're looking at what's called the genome. The genome, or kind of, yes, we are, and yes, we're not. I mean, no, we're not. So the genome is actually the entire genetic code of an organism. And the entire genetic code, we cannot see the code when we look at these chromosomes. We're not looking at the code. What we're looking at is the places that carry the code. So it's like, imagine this is a city. And so you have street number 1, street number 2, street number 3, street number 4, etc. Streets all the way up to street number 39. And on every street, there's a left side and a right side of the street. So you have the odd number houses on one side and the even number houses on the other if you're going to deliver a package. So we have, let's instead of saying odd and even, that's how it is in real life for streets. But for genetics, we're not going to make it odd and even. We're just going to go 1, 2, 3, A, B, C, D, E, F, G, or let's go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and we're going to go all the way down to maybe 500 or whatever. So we have like 500 loci maybe on this chromosome. But we're going to have loci from the female and the loci from the male. So the mother and the father. So every homologous chromosome, these are what's called homologous chromosomes. We have a chromosome from the mother and a chromosome from the father for street number one, chromosome number one. Then we have chromosome number two, one from the mother, one from the father. Then we have chromosome number three, one from the mother, one from the father. And so when we pair up chromosome number three from the mother with three from the father, these are now called homologous chromosomes. The word homo means same, which means they don't have the same genes, but they do have the same traits. So what's the difference between a gene and a trait? A trait is the topic that we're discussing, like hair color, eye color, height, body weight, drive. All right, and maybe it's prey drive. Maybe, and a drive is going to be polygenic, which means polygenics means that there's going to be influencers found on many of these different chromosomes. For example, height is not going to just be controlled by how long your femur is. It might also be controlled by how long your torso is, or how long or how much growth hormone you have somewhere else, or maybe over here you have a gene that affects your metabolism, which might affect your height, or your desire to eat might be, how much food you want to eat might be found on this chromosome. So polygenics is when multiple traits are impacting a single trait, or multiple alleles, multiple addresses are impacting a single trait. That's polygenic. Monogenic is when a trait is found only on one loci. Let's say something is found on chromosome number 31 and it's found nowhere else. Maybe the trait would be, do you have freckles for human beings? Okay, for human beings, though, we can't go over 23 because we only have 23 chromosomes. So maybe the freckles, do you have freckles or not? It's found on chromosome 15. Well, it won't be found anywhere else in this entire karyotype if it's monogenic. There's no other factor that influences it. If it's polygenic, it's going to be found on many traits. Well, coat color is what's called a polygenic trait. So we might have one loci, let's say on chromosome 8. Loci, think of a loci as a location. It's a gene's address or a trait's address. So we have the black or the fawn on one chromosome. Let's just pretend it's on chromosome number 8. I don't know what chromosome it's on, but it's going to be found on one location, one address. You're going to have one trait one gene from the mother and one gene from the father, and they're going to pair up side by side because these homologs carry the same traits, but they don't have to have the same genes. So you could be big B, black, or you could be big B, little b, which is a black masking or blocking the recessive fawn, which is little b. Or you could be little b, which is fawn. All right. So this big B would be what's called homozygous dominant. Big B would be called heterozygous. And little b would be homozygous recessive. Now, think of a lot of people tend to think that if they see a trait, it's dominant. That is not true. Seeing a trait just means the trait is expressed. So let's just pretend that the phone is a dominant trait and my face is a recessive trait. What I'm showing, if I'm homozygous recessive, you're only going to see this appearance, this look, when the dominant trait is not present. If you have a single copy of a dominant trait, it blocks what's hidden behind. Now, what if there was two phones? Well, if there was two phones, my face would not even be present. All you'd have is two copies of the same gene. A dominant trait means if you have one copy, it completely dominates the recessive trait, and it looks exactly the same as if you had two copies of the dominant trait. Because dominance means we're going to block the recessive. It masks or covers up or hides the recessive trait. Fawn is a recessive trait to black. Now, going back to this picture, so let's just say hypothetically that we have a big B and a little b. Well, a black dog could, when it makes its sperm or its egg, it could throw that big B that it got from its mother or father to the sperm or the egg, and let's just say it threw the big B. Well, that means the puppy is going to be black, right? And then you won't be able to produce a fawn dog from that one if it gives the big B. But if it has a little b, then it could throw a little b. So even though this dog would be black because it's big B, little b, it could produce a recessive offspring that's fawn. But if there was two little f's, If you have a fawn dog and there's no big B present at all, the only way it could have a black offspring is if it was bred to a black dog that produced a black that threw the big B. Because if you have two fawn dogs, that means you have two little Fs. If you are two little Bs, and when you do a Punnett square on that, let's see if we can pull up a very simple Punnett square. All right, so I'm going to pull up a Punnett square, and here we go. So let me share this tab. So here we have, let's say, a heterozygous. Big B is black, little b is fawn, big little b. So we're going to breed a black dog with a fawn dog. So this black dog could produce black puppies. This fawn dog cannot. This fawn dog can only throw fawn dogs. The only way you're going to get a black dog out of this parent is if you breed it with a black dog. The black dog is responsible for producing this big B, not this parent. So what happens if you breed two homozygous recessives? So I'm going to show you homozygous recessive. Homozygous means the zygote, which is a fertilized egg, has a copy from both parents that are recessive. All right, so we're going to look at a Punnett square of homozygous recessive. All right, so these are not Bs, but it doesn't matter. We can pretend that these are Bs and look at, well, let's look at this one. All right, so if this was a little b, a fawn dog, and this was a little b, a fawn dog, and we bred them together, then all of the offspring would be fawn. But what if, like I said, what if you actually had a chocolate dog and not a black? This would be a good example of that. No, it's not. So this is what's a polygenic trait. We have two loci, a loci for the C and a loci for the P that's affecting the outcome. And so chocolate is a modifier. So let me look up what's called a dihybrid cross. And we're going to look at so here's a dihybrid cross. In this case here, you have, let's just say the Y stands for big B and the little y stands for little b. So we have black bred to fawn. And then we have non-modified, which the R, let's say, is non-modified, and little r is modified. So we have a fawn dog that carries the gene to modify the black to a brown, but there's no black present, so this modifier is not expressed. So that means if we had a little r with a big Y, you would actually have a brown dog. And so we're going to look for the second generation, and that's going to be right here. So now we're going to look at the second generation. So we have, this is going to be the children that are produced right here from that one I just showed you. So let me back up if I can. I don't know if I can click back. I don't know if this just going back is going to show you the other. Yeah, it did. All right, so here we have a homozygous. This is a homozygous dominant parent. It's carrying Y copies and it's carrying R's. It does not have any little R's. So no matter which combination, we're going to show you this other picture here. Oops, I'm sorry, I'm not sharing the right tab. So what I was showing is, let's say that the R and the Y that we were looking at on this other picture are, let's say the Y's are found on chromosome 1 and the R's are found on chromosome 2. So we have to look at the combinations. We could get this first chromosome for number 1 with this first chromosome for number 2, which would be big Y and big R. Then we could also get this first chromosome with that second one. You're only going to get one of these when you make an egg, and one of these when you make an egg, or one of these when you make a sperm, and one of these when you make a sperm. Now, there is something called crossing over where they can split halves and trade parts, but we're not going to get into that right now, because in either case, when it comes to the Y, you're only going to get one of the Y's and one of the R's. So no matter the combination, what we're going to have is big Y, big R, big Y, big R, big Y, big R, big Y, big R. And the reason that's the case is because this is big Y, big Y, big R, big R. In other words, this individual would be homozygous dominant for those traits. And so if the Y's are black and the R's are modified to brown or not modified to brown. In this case, the big R would be not modified. What that means is this dog is going to be black with no modifier. And since it's homozygous, meaning the two Y's are the same and the two R's are the same, and it's homozygous for both traits, all it can produce is a dominant big Y, big R, no matter which of the four combinations you get. First to first, first to last, last to first, last to last, meaning the first chromosome for number one with the first chromosome for number two. The first chromosome for number one with the second chromosome for number two. The second chromosome for number one with the first chromosome for number two. Second chromosome for number one with the second chromosome for number two. They're all, no matter which combination we get, they're all an unmodified black. Somebody said you need to draw blood to know. No, you don't. There's some tests that you can do to see what's going on. Okay, and by tests I don't mean blood tests. You can actually look at your population and your pedigree and figure some of this out. Depending on if it's a monogenic trait, you can figure it out. If it's a polygenic trait, it's going to be a lot more difficult. But we're going to just continue with monogenic traits for just a minute. So now we're going to look at these four. So dominant R was the non-modifier. Little r is the modifier. So this one, non-modifier means it does not modify the black, and little r means it does modify the black. But in this particular case, we don't have black. We have fawn. So in other words, this modifier is not even going to show up. You're carrying the gene, but you don't even know it. All right. So we have a fawn dog that's going to turn the black to a brown or a black to a blue, depending on which. Let me go to this tab, share this tab instead. So let's say we have the black or fawn on this pair of chromosomes. We have the modifier of black to brown on this chromosome, and we have another modifier on this chromosome that turns black to blue. Well, what happens if we get a black dog with the chocolate modifier and the blue modifier at the same time? Well, this is what's called polygenics, and what the result would be what's called a tawny dog, or also called a champagne. So if you have a blue modifier that turns black to blue, that's just going to be a blue dog. But if you have a brown modifier that turns black to brown, that's going to be a chocolate dog. But if you have the chocolate modifier and the blue modifier at the same time while also having a black dog that's big B, or big B, little b, now you have a champagne dog or a tawny dog. What if this was fawn? If this was a fawn dog, little b, then the chocolate modifier would not matter and the blue modifier would not matter. You wouldn't even know that you had it. All right, unless you did a lot of breedings and you were able to look at the population, look at the pedigree, look at the offspring, do a test cross, which means to breed with a homozygous recessive. So you can do a blood test if you want to, or you can do what's called a test cross, which means breeding with an individual that cannot block anything. Remember, I had said that, let's go to this screen. So I said a dominant hides or blocks a trait. So that's going to be a dominant gene is blocking something that's recessive underneath. Well, if a test cross means we want to see what's going on because we're testing. In other words, you're breeding to an individual that has no blockers, no dominant traits, no masking. And a lot of people will look at like red nose, and they will think that a red nose is dominant because if you breed a red nose to a red nose, you only get red nose. And that while that's true, that actually means it's recessive. It's recessive, but the allelic frequency is high. Now, what's funny about canine genetics, and we're going to go back to, let me move this here. So if we go back to, oh my goodness, come on. All right, there we go. So we're going to go back to this screen. We have a black fawn, we have a chocolate modifier, we have a blue modifier, and let's say red nose is on this one. Red nose, if you have a dominant and a dominant, that means we're going to see everything that we have over here. If you have a dominant recessive, this dominant means show what we have. For the red nose, if we have any dominant genes, we're going to show what we have over here. If we have two recessive alleles, which is the red modifier with the red modifier, red nose, when you have two of these, even though it's recessive, a unique thing happens. And what happens is it turns off all of these genes. So a recessive condition on another chromosome can impact the expression or lack thereof for other chromosomes. So that's what's called polygenics. So two red nosed dogs is a recessive condition, but ironically, in this recessive condition, we're going to block dominant traits. That's very unusual. That can only happen basically when you're dealing with polygenics, and polygenics means a trait that's influenced by multiple loci. So back to a better understanding of what this picture means. Think about these as different streets in a city, and each street has address number 1, which is going to be A and B. Address number 2 are mother and father. Address number 1 for the mother, address number 1 for the father. Address number 2 from the mother, address number 2 for the father on the street number 1. And we're going to go all the way to maybe 500 addresses just on street number 1. So 1 through 500 from the mother and 1 through 500 from the father. Then we're going to go to the next street, and the same thing's going to happen, but this street's a little shorter than that street. So instead of having 400 addresses here, we might have, instead of having 500 addresses here like we do over here, we might only have 450. And these chromosomes are listed, you'll notice, from longest chromosome to shortest chromosome. So down here, we might only have 50 addresses on this chromosome. So these addresses are called loci, which is the location of a trait in the genome. Now, over here, we have something unique. People a lot of times ask which parent has the greater influence on the offspring, the mother or the father? And basically, it's 50-50. So, but technically, the mother has slightly more influence when you have a male offspring. So if we were to look at this here, we have an X and a Y. X and a Y means we have a male offspring. By having a male offspring, I want to make sure I'm showing you the right screen. Oops, yes I am, but let me need to minimize myself. All right, so by having a male offspring, we have an X chromosome from the mother and a Y chromosome from the father. You'll notice the amount of addresses that we have on this Y chromosome are very, very small. So as a result of that, that means the mother impacts male offspring slightly more than the father, but we need to keep this in perspective. It's very disingenuous to say that the mother impacts the offspring more than the father, because in the bigger picture, that's not really something to worry about. And the reason why is, let's add the length of all these mothers. Let's just pretend the first one is from the mother on each one of these chromosomes. If we add all these up into a single strand, then let's say the mother's chromosome, all of her chromosomes might go across this screen and back. Okay, so the mother's chromosomes are long enough to go across this screen and back if we put all of these pieces together. Well, if we do the same thing for the father, okay, his is going to go all the way across the screen and back just to about right here. Why? Because the distance from here, I'm assuming y'all can see my mouse. It's only, if we look at all of this, it's only missing a segment, a small segment of the total DNA. It is missing a large segment for this chromosome, but this chromosome is only one of 39. So the Y chromosome has genes that are missing for a male offspring. But if this was a female offspring, that would be two X's. There would not be a Y chromosome. An XX makes a female. So that means you have a full set. So the mother has no more influence on the females than the father. The father is just as influential on a female as the mother is. The only time a mother has more influence on the offspring is if the offspring is male. And the difference is very minute. So people say, who's more important, the male or the female? Well, the female may be more important for a couple of factors, but it's not a major concern. All right, it's more important. A female is more important than the male if you have a male offspring, but only a tiny bit, as I showed you here. The other thing that the female does is she provides the mitochondrial DNA. And since the female provides the mitochondrial DNA, she also has a slight tiny bit more impact onto the offspring than the male does, because we're trying to get that sperm into that egg. It's a race. There's millions and millions of sperm racing to get to that egg, and if they're carrying a bunch of mitochondria, which is DNA that's not in the nucleus, okay, all of this is called nuclear DNA. But the DNA that's not in the nucleus, it's in the cytoplasm in a structure called the mitochondria. Um, if it has a bunch of those, that sperm has to carry extra weight, and it's going to probably lose the race to fertilize that egg. So, and plus, the sperm needs to be small enough that the sperm's tail can swim and push that sperm to that egg. So it has to get there, and it has to get there first. Then it has to go through something called an acrosome reaction, where the head of the sperm, the tip of it, dissolves and penetrates that egg, and it deposits that DNA into the egg. When it does that, the egg, the outer shell of the egg, loses the receptors of the sperm. So when those receptors fall off that egg, all the other sperm are now chasing receptors that are no longer attached to the egg. Imagine you're playing baseball and somebody's throwing a ball. The ball is like the sperm, and the glove is the receptor. Well, after that first egg, or excuse me, after that first sperm gets caught, that first sperm gets caught by the egg, the entire field, let's say you had all of the baseball players in the whole country, they drop their gloves. When they drop their gloves, the balls are still chasing the gloves, but the gloves are not attached to the egg anymore. They're not on the baseball players anymore. So as a result, none of those sperm get into the egg. Only the first one. That's when if two sperm ever get into an egg, that's called polyspermy. And instead of having two chromosomes for every location, you're going to now have three chromosomes, because you had one from the egg. One of these chromosomes for every one of these addresses comes from the egg, and one of these chromosomes comes from the sperm. If two sperm come in there at the same time, which is an extremely rare situation, the baby's going to die, because it's going to have three sets of chromosomes instead of two. Now, it's always going to be dead. There's going to be no cases where polyspermy works. Now, if you had that happen on one chromosome alone, just one, let's say it happened only on chromosome 32. For humans, this can happen on chromosome number 21. We have 23 chromosomes in a human being, and if we get not polyspermy, but an extra chromosome, that was actually not polyspermy for chromosome number 21. That was in gene segregation, when the chromosomes were supposed to come apart during meiosis. They did not come apart. Both of those chromosomes in the mother, when she was making her egg, one was supposed to go to one egg formation and the other was supposed to go to another egg formation, but they made a mistake and both of them went to the same egg. And then the other egg over there didn't get any. All right. Because at cell division, we're making multiple eggs during gamete formation, which is the production of sperm or egg. Well, if both of those chromosomes go to the same egg, we're going to have an extra chromosome. And in trisomy, this is called trisomy. Tri means three. So that baby that's born is going to have three chromosomes. And in the case of trisomy 21, where you get two from the mother and one from the father, you have an extra chromosome. That is not good. It causes a disease known as Down syndrome. Down syndrome causes mental retardation, a thickening of the tongue, a shortening of the neck, and it's a condition that's genetic. Another location that you can get trisomy 21 trisomy, not 21, trisomy 23 is called Klinefelter's. Or you could have triple X, which is when you have three sex chromosomes. Triple X would be XXX. You'd have a female that has an extra chromosome. Klinefelter's is when you have an XXY, all right, something along those lines. And then there's other conditions that can happen as well. So you're never really, in a normal population, you're never really going to have three chromosomes. You're going to have one loci that has a chromosome from the mother and a chromosome from the father. And this is basically how genetics works. Now, on this screen, let me go back to this tab. So let's say we take this individual right here, heterozygous. It's got one copy of the black and one copy of the fawn, one copy of the unmodified, one copy of the modified. What happens if we breed this individual with this individual? That's what's known as a dihybrid cross. Okay, so if I took out the word recessive, we would get, in fact, I'll do that real quick. A dihybrid cross is going to look like, here we go. So this is a dihybrid cross. In a dihybrid cross, we have all these combinations. Remember, I want one that shows you the alleles. Give me just a minute. Let's find one that shows you what the parent's genotype is. Still not right. So this is not a dihybrid cross. This is a dihybrid. Big T, big Y, little y. This is not a dihybrid because we're heterozygous for the T's, but we're not heterozygous for the Y's. I'm trying to find one that has different T's and different Y's for both parents. See how it says heterozygous for the T, but homozygous for the Y? That means it's different T's, but same Y's. Hetero means different, homo means same. I want one that's heterozygous for both. That's hetero for both. This is homo for the other. All right, so here we have, all right, so this one's a dihybrid. We have a parent one is big A, little a, heterozygous for A's, and heterozygous for B's. So what this means is, let's say that this is black and this is modifier, brown. So here we have black unmodified. Big dominates means unmodified blocks the modified. Okay, so we have a black and it is black. We have a black dog and we bred it with another black dog. Black and not modified. Well, when we breed a black with a black, what we can see is we can actually produce a number of unique combinations if they're heterozygous. So we have black paired with black, which means we have a black. And we have unmodified with unmodified, which means we have unmodified. Now, if we come over here, we have a black bred with a black unmodified that this parent was able to, I like to write first to first, first to last, last to first, last to last. First to first. That's right here. First to last. Big A, little b, right here. Last to first. That's the last A with the first B right here. And then we have last to last. Little a, little b right here. So what happens if we get first to first from this parent with last to last from this parent? Means we have big A, big B with little a, little b. Well, what we're going to end up with is still a black dog that's not modified, because the black is going to block the fawn and the unmodified is going to block the modified. However, what happens if we get last to last bred with last to last? So we're going to have last to last bred with last, or the sperm produced from this male, last to last, is combined with the egg produced from this female, last to last. So we have this sperm provides last to last to this egg that provides last to last. Now we end up having a black dog bred to a black dog that produced a fawn dog. All right. What if we had this one here? We have a black modified bred with a black modified. Now we have a black dog that's been modified to be brown. A lot of people will confuse this with a fawn dog. It's not a fawn dog. This is a chocolate dog. Okay. And so if we take this combination, big A, little b, homozygous for one, homozygous dominant for one, and homozygous recessive for the other, and we breed it with a fawn dog, little a, little homozygous for recessive for both. So let's do that. We're going to try to find a homozygous A and heterozygous with homozygous recessive, homozygous dominant. I don't know if this is going to show up really well, and we need a dihybrid. See, if I could share you my own screen instead of what's online, I could just make this problem up, but I'm not able to share what's on my own computer. I can only show you what I find online. For some reason, StreamYard limits us to that. Let's just use this one, or let me find a blank one, a blank, and maybe we can fill it in. All right, so we're just going to take one. one of these. This may be it. Let me look at this and see if this is what I need. So let's say we have a black dog that's modified to chocolate. So that's going to be our black gene, and that's going to be our modifier, chocolate. So we have a dominant black with a chocolate modifier. And all these are going to be, no matter the combination, we have a black dog with a chocolate modifier. So every single one of these eggs is going to be black with modified to brown. Over here we have fawn, but this dog carried modified and non-modified. So as a result of this dog being a fawn, we didn't even know that this dog was heterozygous. Okay, so it'd be we're going to show the black gene when it's present, but we don't even know it because we don't have the black gene present. Here we have a fawn dog. So in this combination, we now have a big G. I wish I could write on this: big G, big B, little b. Now, what we have now done is we've non-modified the blacks. We have a black puppy. Here we have a big G with a little g, so we're going to have a black dog, and the big B is going to be non-modified. So we still have two black dogs. Now we have, in all of these cases, actually, we're going to have a black dog. In all of these cases, we're going to have a black dog, and it's going to be non-modified. So all 8 of these squares are going to be black dog, not modified. But down here we have something different. Even though this is a fawn dog, this fawn dog cannot produce black, but this one does, and this dominates that. So same thing down here. We have the fawn is going to be little g in all 8 of these, but we're going to get big G in all 8 of these from the other parent. So now all of these are black, but because there's a modifier, and no matter which sperm we get, all of these are modifiers. And no matter which egg we get, these are modifiers. So now we have a modified black dog, which means this dog is going to be chocolate. So if they had 16 puppies, the odds are 8 of them would be black and 8 of them would be brown. Now, that doesn't mean that's what you're going to get. That's the odds. Like that when you flip a coin, the odds of getting heads is 50-50. That doesn't mean you're going to get heads 50% of the time. It just means that's what you should expect to get. But if you flip a quarter one time, you're only going to get all 100% heads or 100% tails. If you only had one, you can't get both at the same time in the single head coin toss. You're going to get only heads or only tails, but the odds are 50-50. However, if you flip that quarter twice, you might get heads, you might get heads, tails, you might get tails, heads, and you might get tails, tails. So here's heads, here's, let's say heads, let's just go to a blank one. Here's heads, here's heads, tails, here's tails, heads, and here's tails, tails. All right, so the odds are now 1 out of 4 times if we went to a monogenic. Here we go. So let's say this is heads and that's tails, this is heads, this is tails. Heads, heads, tails again, but it actually was tails, heads. So we have this, we're going to read this letter first and this letter second. So the mother produced a heads, the mothers produce a heads. The father produced a heads, the father produced a tail. So we have heads, tails. But now the mother produced a tail and the father produced a heads. So we're going to actually have tails, heads, but we're going to write heads, tails because we always write the dominant first. And then here we have tails, tails. So what happens when we have 4 tosses, it is possible that we get heads twice. It is possible that we get tails twice, but we also expect 50-50. All right, some of them are going to be mixed. Now, in this case, the dominant is going to hide, the heads is going to hide the tails. So we wouldn't even know the tails was present if this was genetics. On a coin toss, you're going to know it, but in genetics, you're not going to know it. Okay, in genetics, the dominant gene is going to hide it. All right, let me go back to our screen that we were before. Okay, on this Punnett square here. And so now let's just pretend that we had this individual here was not heterozygous. let's say it was big G, little b, the same as down here. Now we would have a brown dog because the modified of the black is all we can get. And we'd also have a fawn that's modified, but we don't know she's modified because we don't have a black gene to turn brown. We only have a fawn dog. So if we have a fawn dog that carries a modified, and we have a black dog that is carrying the modified, then what we would get down here is all of the puppies would be black modified to brown. All of them would be big G, little b, in these squares. And if we had the same thing up here, then we'd have 100% of that combination. But here we have something different, so it's a 50%. I hope that's clearer than mud. I don't want it to be so hard to understand. Hopefully that makes some sense. So this is just some basics on genetics. We still haven't discussed a lot of different topics. Let me share one more tab. I'm going to recommend that y'all read the description to this video, because basically what I have done is I've only introduced the field, okay? And I'm going to share this tab. I have noticed fake it till you make it. The mindset has contaminated the dog market. There's a lot of people in the dog market that are saying genetics and they don't understand it. They're not an engineer. They're not a mechanic. They're not a master mechanic for sure. A lot of them are not even a mechanic. Some of them can't even change the oil. All they can basically do is put air in the tires. So those people, if you can only put air in the tires and you really don't understand a lot of these concepts that I have described right here, all right, if you don't understand what a diploid cell is or a haploid cell is, if you don't understand what gene segregation is, if you don't know what that fertilization actually means genetic recombination, where two haploid cells create a diploid zygote, if you don't know that a zygote is the first stage of the offspring, if you don't know the difference between a monogenic trait and a polygenic trait, or what dominant and recessive truly mean, this is one that I see mixed up a lot. A lot of people think that dominant means expressed and recessive means not expressed. While that's true, that's only the case in a heterozygous individual. It means you have one copy of each. You don't know if a trait is dominant when it's by itself. If it's homozygous, you don't know if it's dominant. All right, you just know it's expressed. Is it expressed because it's dominant, or is it expressed because it's got a high allelic frequency? So don't confuse dominant and recessive with allelic frequency. What's the difference between a trait that has a high heritability index versus one that has a low heritability index, which means it's influenced by the environment? Some traits, like hip dysplasia, are not only genetic, they do have a genetic factor, but they're also influenced by the environment. Same with how much your dog weighs. Your dog's weight is influenced by its genetics, but it's also influenced by its exercise, what it's eating, and things like that. So heritability index is going to be influenced by genes and the environment. The lower the number, so heritability index is reported from 0.0 to 1.0. If it's a 1.0, that means it's completely controlled by genetics and the environment has no impact whatsoever. That's going to be something like basically coat color. Okay, the environment's not going to change it on a dog. Then you're going to have something like, for humans, maybe freckles, or do you have a dimple in your chin? These are genetic factors. They're not environmentally influenced. But body mass, how much you weigh, how much muscle mass you have, these are influenced by the environment. Learn these basic concepts before you get on YouTube and try to talk about genetics. It's very frustrating to me when I see these people talk, I breed for desirable traits, and I'm like, you can't even do a Punnett square on a monogenic trait that's simple inheritance. They don't understand it, much less trying to get into calculating the coefficient of relationship, or which is properly abbreviated as RC. I and many others oftentimes say COR because COR, coefficient of relationship. But the proper abbreviation is not actually COR. The proper abbreviation is RC, relationship coefficient. and the coefficient of inbreeding, which is COI. What's the difference between these two? So a brother and sister have a 50% coefficient of relationship if they're a full brother sister, but their coefficient of inbreeding could be 0. Um, so if their parents are unrelated. Um, Gain an understanding of the difference between an individual's genetics and population genetics. Individual's genetics is going to be dealing with monogenics, polygenics, dominance, recessive, and whatnot. Population genetics is going to be dealing with allelic frequency. How often does a certain trait exist in a population? And if a recessive gene has a very high allelic frequency, people will oftentimes think it's a dominant trait, but it's not. It's just a trait that's really popular in the population. Let's take an example with human beings. If you have a white person from Sweden that marries a black person from Africa, and their ancestors have all the Sweden persons are all from Sweden, blonde hair, blue eyes, and the African persons is all from Africa, darker pigment skin, black hair, and etc. If they breed them together, you're going to see not dominance, not recessive. You're going to see what's called incomplete dominance. The child would partially express traits from both parents. They're not as black as an African from Africa, and they're certainly not as white as a white person from Sweden with blonde hair and blue eyes. They're somewhere in the middle. That is an incomplete dominance. However, what happens if you had something where you had, let's say, stripes? What if some parts of that body were black, just as black as a black person from Africa, and other parts of the body were just as white as a white person from Sweden? Because you could see the white in its pure form in that mixed individual and the black in its pure form in that mixed individual, that would not be incomplete dominance. That would be what's called codominance, meaning both dominate in different parts of the body. All right, so you have to understand the difference between incomplete dominance and codominance. The Hardy-Weinberg equilibrium equation allows you to measure allelic frequency in a population. The founder effect means the individuals that you use to start your breeding program, you need to know the genes that those dogs carry, and you're going to get an understanding of that by looking at the traits of their ancestors. What are the steps of natural and artificial selection? If you don't know that, you don't need to be making videos about genetics. There's basically 4 steps in natural selection. The overproduction of offspring, variation exists in the population, competition for resources, meaning they're going to go out and compete for those resources, is the one that find those resources are successful, and the ones who find them reproduce at a higher rate. So those are the 4 steps of natural selection. In artificial selection, the first 2 steps are the same. Overproduction of offspring and variation exists. But the third step is different. Now, artificial means we're domesticated animals. They're no longer in the natural world. They live in an artificial world because we are going to control whether they get the resources. We are going to control whether they survive. They're not going to have to compete for the resources. Instead, they're going to compete to please us. They're going to serve us or give us what we want. And if they give us what we want, then we will provide for them and allow them to be bred. They don't breed on their own. We're going to choose the breeding. So for artificial, it's overproduction of offspring, variation exists, competition to serve or please, and the ones who serve or please the most are bred. Bottleneck is when you have a population that has many different genes, but you're going to select for something in particular, and you're only going to let those genes go through the funnel. So the funnel is the bottleneck. We're going to let the desired traits come through to make the next generation, but the previous generation with the undesired traits are selected against. So bottlenecking is a culling process. Genetic drift occurs through, excuse me, genetic drift occurs through bottlenecking. By eliminating undesired traits, we're going to increase the allelic frequency of the desired genes, but decrease the allelic frequency of the other undesired genes and purify the new population by eliminating the undesired traits. So slowly over time, it's drift because it's very slow. And over time, we're going to make the desired traits higher frequency and the undesired traits lower frequency. Gene flow, think of a river that flows. If you have a boat that's in a river, the boat's going to move very quickly because the river is flowing. If you have a boat in a pond and there's no movement of water, you're going to just drift very slowly. So bottlenecking genes occurs very slowly, but um, I had a beep on my phone here. Let's see, or on my messenger. I don't know if somebody's trying to send me a message. I'll check real quick. So here you're going to drift real slowly because it takes a long time, generation after generation after generation, to change the genes. But gene flow is very quick because this happens from an outcross. So gene flow is from an outcross. Genetic drift is from selective breeding. We need to learn these basics before we start making videos on that. I'm going to go to my screen share page real quickly and see if y'all can still able to hear me. I'm blowing today, Lee. You're really excelling this. Well, I appreciate that. I hope I'm going to come back through these questions when I get done. Inbreeding, which is like a father, daughter, mother, son, or full sibling, can be done to accelerate the process. And we're talking about accelerating the process of genetic drift. So we're going to, where's the genetic drift? I had that right up above. Oh, okay, there it is. So I went too far. So line breeding combined until then. All right. So here's a message to the fakers. If you don't understand these concepts, do our community a favor and stop speaking as if you're an authority on it. Sometimes people know enough to be dangerous. And look, we all make mistakes. I'm not saying that you can't speak if you have a good understanding. If you have a good understanding, go ahead and speak on the field. But one of the problems with social media is somebody can come out here and ask a sincere question. They might say, hey, I noticed that my dog has this sore. What should I do about it? Well, the problem with that is they're going to get a lot of people answering the question. Maybe the dog got bit by a snake. What should I do about it? Well, yes, you can get information very quickly that might be important to save your dog's life if it got bit by a snake. However, most people are going to give you bad information. And right now, you don't need bad information. You need good information. So call your vet, okay? Call an expert. If you don't know who's an expert, now, if you know an expert, call an expert. But don't get your information off social media for some of these topics from people that you don't know that can prove and verify what they're talking about. They might tell you to take an aspirin, all right? Give the dog an aspirin or take a Tylenol, which is, you don't want to do some of these things because some of these medications are safe for people and some of these are safe for dogs and some of these human medications are not safe for dogs. They might tell you to give the dog a Benadryl. Benadryl might help with a snake bite, but it kind of depends on how many times the dog was bit and where was it bit. If it was bit in the throat and the throat's going to swell up and cut off because the swelling might cut off like a chokehold on the jugular and reduce the blood flow to the brain, maybe Benadryl will help, but maybe it won't, especially if it was bit multiple times. Maybe dexamethasone is what you need. Okay, but a lot of people don't know what dexamethasone is, or if you don't have it, how are you going to get it? So be careful of the advice that you get online. There's a lot of fakers till you make it. And I don't like fakers till you make it because they do a disservice to the community. Because if you're ignorant, you're trusting someone that's calling himself an expert to be an expert, and they might not be. All right. Now, again, everybody can make mistakes. I'm not trying to rain on anybody's parade, but there's a very popular equation that I've seen out in the dog world that really is incorrect. And I'm disgusted when I see it because it's not backed up in any genetics research. It's just something that dog breeders have made up. And I am going to have a video about it. And I've actually tested the equation, and sure enough, it does not work. It's a bunch of nonsense. And there's a certain person out there that claims to be a canine geneticist, I guess you could say, that promotes this equation, and it's just incorrect. It doesn't work. But I'm going to save that video for another day because I need to go in and explain why that doesn't work. Line breeding, misinformation. So just stop it. Stop selling snake oil. These people are calling themselves geneticists because they want to sell their puppies and they want to sell snake oil. They want to sound like an expert, but they're only at the level of being able to put this mechanical level of putting air in their tires. These guys, so many of these people are not even at the level of changing their own oil or changing their air filter. They're just not at the basic level. And even if you are at the basic level, you're still not a mechanic. And even if you are a mechanic, you're still not a master mechanic. And even if you are a master mechanic, you're not an engineer. So some information when it comes to how a car works, some of those topics need to be discussed by engineers. Some of those topics need to be discussed by master mechanics, because there's things that master mechanics can do that an engineer cannot. A lot of engineers are nerds, so to speak, that can't even turn a wrench. I saw a video with a movie with Billy Bob Thornton called The Landman. He put a pipe wrench on a nut to turn that nut, and he had the pipe wrench backwards. You know, if you've ever used a pipe wrench, you push it one way and the wrench closes, you push the wrench the other way and the wrench opens. So he's turning this bolt and he's pushing it where the wrench is opening, because he's pushing the wrench backwards. I'm like, don't try to pretend like you're an oil man if you don't even know how to use a pipe wrench. Okay, so it's just kind of silly when you see something like that. I noticed it immediately. I mentioned it to a friend of mine that's in the tree business. He's like, yeah, I saw that too. I mean, you got this guy trying to pretend to be a rancher or pretending to be an oil rigger, and he can't even turn a pipe wrench because he's an actor in real life. All right. And that's the way some of these dog breeders are. They're actors in real life. Line breeding combined with performance selection and health testing should be, for the most part, the primary practice for good breeding programs. Meaning, if you don't understand genetics, you can still be successful. You just need to have good selective breeding. And that's going to get you 90% of the way. All right. And line breeding is when you're going to focus on a single individual in a population, but it's not necessarily super close. So you might be focusing on a grandparent or a great grandparent, or breeding, descendants that are grandchildren from that same grandparent back to each other. You're breeding cousins together, basically, or half siblings. Something like that would be line breeding. Now, truthfully, in genetics, and this is going to have some of the canine people ruffle their feathers, line breeding is inbreeding. People are like, no, it's not. No, it's not. It's looser. You're right. It is looser. But when you talk to geneticists, if you have father, daughter, mother, son, full siblings, yes, that's inbreeding. But cousins is inbreeding also. It's just loose inbreeding. And in the canine world, we call it line breeding. In the horse world, they call that line breeding. But in the genetics world, it's still inbreeding. Okay, it's just not as tight. Okay, so when people say there's 3 types of inbreeding, that's true in the small community of dog worlds: father, daughter, mother, son, full siblings. And line breeding is going to be many things. It could be uncle, niece, aunt, nephew, grandparent, grandchild, or cousins. But in the actual genetics world, and not in the canine world, this is just a looser version of inbreeding. All right. Next, outcrossing is done for 3 reasons. Inbreeding is done for basically 1 reason. Inbreeding is done to make the population consistent. Outcrossing is done for 3 reasons. Either the line is so tight that you want to get rid of inbreeding depression. Inbreeding depression means your fitness in your population has declined because you don't have heterozygosity in your line. So you have too much homozygosity. Homozygosity is when all the genes are starting to become the same. The homo means the same, and zygosity refers to the zygote, or the egg or the baby. All right, excuse me, the fertilized egg or the baby. So a fertilized egg always has 2 genes. If those genes are for every trait or every loci, if those genes are the same, they're homozygous. If the genes are different, it's heterozygous. Some traits are controlled by multiple loci, which means polygenic. And gametes is a trait that's polygenic. People say gametes is recessive. That's not correct. Gametes is polygenic. It's not controlled by one loci. If it was, it'd be very, very, very easy to reproduce time and time and time again. And we know for a fact that you can have a population of game dogs and breed them with a population of game dogs and still end up with curs. So that means it doesn't breed true like people think it does. It can, when you breed game with game, you increase your chance of getting game, but it doesn't guarantee it. And that's because there's other things that are coming in that we may not see on other genes. We might need a degree of heterozygosity on some of these polygenic traits to affect it, and that makes it harder to select for it. Inbreeding depression is one reason to outcross. Another is because you see a trait or line area that could be improved. For example, let's say your dog is excellent in some areas, but not necessarily all areas. And so you're going to bring in something that you desired. That would be a reason for outcross. All right, we have this in this description video. Now, I made this I selected this picture kind of as a joke for this video because sometimes I see people, man, this happens all the time. There's a person that just asked me this recently, and he might think that I'm talking about him. I am not. This happens to me all the time. They say, hey, which puppy is standing out? Which one do you like the best? And I'm like, the puppies are 3 weeks old, buddy. They're the size of rats. I don't know the difference of one and the next. So I can't tell you which puppy stands out until they're older. All right. And then you see somebody that buys a good prospect at 8 weeks and they're like, dude, this is going to be a great stud. You don't know that. The dog's 8 weeks old. Let it grow up. See if it has hip dysplasia. See what its drive's like in different environments. See what its nerves like. See if it's healthy. You don't know if this dog's going to be healthy yet. You got to let it grow up. And so I've jokingly used this picture before and said, hey, this is one awesome stud right here. Stud fees, $2,500. Well, the puppy's not even born yet, and they're talking about how they're going to breed it. Take your time, slow down. It's not a race to the next dog. Breeding dogs is not a sprint. It's a marathon. It takes time, and over time, you're going to slowly get better through gene flow and genetic drift. All right, let me close this tab. We're going to go back to the StreamYard picture, close this tab, and now I'm going to look at some of the questions and see what y'all had to say. Hey, if y'all like this video, please like it and share it. I'm not good at the promotion side. I'm not good at getting my algorithms up because I don't ask my viewers to like and share. Please like and share. If I'm giving you information that you appreciate, do me a favor and like and share. I need to grow this channel. I need to promote truth out in the industry. I can speak on what I know. Here's how I speak. If I know something, I'm going to tell you it's so. If I think something, I'm going to tell you I don't know, but this is what I think. And then if I have no really great opinion on something, but I kind of have an opinion, I'll tell you, well, I don't know, and I'm not even sure, but I would guess this. I'll tell you, I know, I think, I'm guessing, and then sometimes I'll say, look, I don't have a clue on that one. I'm just going to defer to somebody else, okay? So that's what we need to do as responsible members of our community. If you take your car to somebody that says he can fix it, and this is a huge problem now in this world, the fake it till you make it is costing innocent people, in some cases, times hundreds of dollars, sometimes thousands of dollars. If you take your car to a guy that says he can fix it and he takes it apart and he puts it back together and he doesn't put the drain plug back in when he's supposed to fill the oil and he didn't do it right, that costs you thousands of dollars, and you hope that you can prove that he did it. And if he's broke because it's not a professional shop, it's just some small podunk private citizen that did it, crackheads are cutting off catalytic converters, or meth heads are cutting off catalytic converters, saying that they're going to give you $50 for a catalytic converter. And I know people that have let them do it. All right, it's crazy that people let somebody do that, and then they find out that they need their catalytic converter. They're like, oh, you don't need it. Like, oh, next thing you know, they got to spend a couple $1,000s to get their car running right because they sold the catalytic converter for $50 because they had no clue what they were doing. If you don't know what you're doing, don't speak on the subject. Speak on what you know. I can learn something from you if you speak on what you know. If I listen to you and you don't know what you're talking about, you can destroy my program. So you guys have a responsibility to protect your program. Good show last night. I thought it was a pretty good show too. Thank you. Melee 20. We talked about this guy as a mechanic. He said he doesn't like engineers, and I get it. We are experts in different fields, and an engineer can do some things that a mechanic can't do, but a mechanic can certainly do some things that an engineer can't do. I've seen engineers that can't even drive a lawnmower. What's good, Lee? What's the earliest age a male could procreate? Technically, a male dog can breed it. I mean, technically, some of them could breed it 6 to 8 months. Anybody that breeds a dog at 8 months old, though, has got some screws loose. Be patient, test that dog, know what you got, and wait. So I would say The earliest I would breed a dog would be the earliest. Let's just, in 24 years of breeding, I bred a dog one time that was 12 months old. And I don't like doing that, but I did it because we wanted to breed that particular dog to an older female that was like already 8 or 9 years old at the time. And I was afraid if I waited, I wouldn't, I'd have to go back and check maybe it was a young female that I bred to an old male, but I wanted to do a certain combination and one of them was on a timeline. But basically, I think that's to be avoided. You can make exceptions once in a while, but don't live off exceptions, because if you live off exceptions and you make that the normal process, you're going to end up losing the traits that you're seeking. So be patient. Oh, he also asked, I know females could be on their first heat. Yes, but if I would also look at that. Let's say you've had a female that had her first heat at 8 months. I'm not going to breed that dog. If she had her first heat at 14 or 15 months, I might breed that dog. So when did they have their first heat? Not everybody has their first heat. Not every female has their first heat at the same age. You don't want to breed on the first heat for a female. On a male, you want to see what the traits are after puberty, before breeding. Okay, I agree with that. Except for it depends. We had a female one time named Snaps. Um, I didn't have her when she was young, but I can tell you from experience there are occasionally females that will very seldom come into heat. And I know of females that very rarely may not have a heat cycle till they're 2 years old. And if that happens, and I like this female, I'm breeding her, first heat or not. If I like this female and it's her first heat at 2 years of age, she might be a dog that has low fertility, and you might think, well, why would you want to breed that? If she's a dog that I like and she's carrying a lot of desirable traits, I'll try to fix the reproductive issues on the next one. All right. So let's get you. I'm hoping that male might provide some genes that fix that reproductive issue. So let's remember the male. If you have a girl offspring, the male produced an X chromosome for the girl offspring. So if I have a female that's got a lot of desirable traits, and I can get a daughter out of her from a male that comes from a lineage that does not have reproductive issues, I can capture the desired traits from the low reproductive female into her daughter that possibly got an X chromosome from the male that fixed that reproductive issue, and I still have a lot of the desired traits that were from the other chromosomes. So I remember, anyway, I was going to say, I talked to about a bitch named Snaps. I bought her as an adult. She had already been in heat a couple of times, probably. But when I had her, I waited for her to come into heat and waited for her to come into heat and waited for her to come into heat. I had her for 18 months and she had a heat cycle. I bred her to 2 different studs. You're like, why would you breed her to 2 studs? Because I can DNA test them and I don't want to wait 2 years again. I can DNA test the pups, DNA test the parents, and find out which pup is off of which parent. And so that's what I did when I bred Snaps. So sometimes you are in a race of time, but you want to minimize that. Don't make that the normal process. And if you do breed 2 studs to a female, you need to be responsible about it and DNA test it. And don't just say, oh, I can look at them and tell. Sometimes you can tell and sometimes you cannot. Like we did the Punnett squares earlier. Sometimes they can throw something that you did not expect them to throw, and you're like, oh, that one looks like the mother. So, or no, that one looks like the male A, not male B. But if male B was heterozygous, he might have been able to throw what looked like male A. So you need to test it and find out which one is the parent in 99% of the times. Now, let's say the one male is blue and the other male comes from a population where there's never been a blue dog ever, ever, ever. I'm not talking about maybe like if you bought a pit bull from the ADBA, from a game dog man like Madison Parker, and for the last 50 years, there's never been a blue dog in that population, and you had a blue female, and you bred that game dog to that blue female, and you got a blue puppy. That blue puppy was not produced by that game dog that you got from Madison Parker. But that's such I'm just giving you an extreme example where you might not have to DNA test. So you have a blue female, and you breed it to 2 males. One is, let's say, a red dog, and one is a black dog. And you have a puppy that is not blue, and maybe the puppy's black. You might think that the black father was the puppy. That might be true, but it might not be true, because the reason it might not be true is that red dog did not carry a black gene, but the blue mother did. Because remember, blue is a modified black. So the blue mother gave the black gene to the egg, and then the red dog did not give a black gene, but fawn is recessive. And since fawn is recessive and red is recessive, now you have a black puppy, and it's not modified, so it's not blue, and you have a black dog. And you think the black father was the parent, but you don't know that because you didn't DNA test it. So you need to DNA test your parents, especially if you're dealing with traits that you cannot visually see and identify. And even if you can identify, does heterozygous and homozygous and dominance come into play? So just DNA test your pups. There's extenuating circumstances. Be careful of the people who say, well, I can look and tell. No, you can't. I agree. Just want to know the earliest. Okay. He's talking about when to breed. Let me scroll through these comments here instead of showing them all. Yeah, you can look for certain diseases that are heritable. No. No. There's red is kind of a complicated topic because you have red, you have buckskin, you have fawn, you have red nose. Red nose is different than regular red. But no, in none of those cases they're not black dogs. Black dog is a dominant, and fawn is a recessive, and then red nose is another recessive on a different loci. What's all right? For the morning, Lee. Thank you, sir. Lee got that game for dogs. Like and subscribe and share, y'all. Yeah. I've studied science a lot, but anybody that claims they know everything is a liar. To me, a geneticist a lot of times is like an engineer. They're not necessarily a mechanic. And what I mean by that is I understand enough genetics to follow along and look at certain things and figure things out. But I would not be what I would call a geneticist. I'm an animal scientist. I understand husbandry. I understand reproduction. I understand some genetics. I understand animal behavior. I understand ethology. I understand Ivan Pavlov and B.S. Skinner. I understand fitness, health, conditioning, nutrition, a lot of these different aspects, but I'm a jack of all trades and an expert of none. The problem why people a lot of times think I'm a genetics expert is because compared to the average dog breeder who knows nothing about genetics, they barely, and their mechanical skills are like compared to genetics, is like putting air in a tire. Okay, they just don't know genetics at all. So yes, I would say I'm like a master mechanic, so to speak, when it comes to genetics, but I'm not an engineer. Okay, somebody that's going to go out and do gene modification through manipulation. And then a friend of mine, I'm going to mention his name, David Ishii. He's kind of like the engineer, but he doesn't, in my opinion, not trying to throw him under the bus, but I'm trying to be realistic. He doesn't have the canine training and handling skills as maybe a dog trainer would. And I think he would agree with that. Okay. So, he's a little bit on the engineer side and a little bit less on the mechanical side. So, because a dog breeder is not just a nerd. I'm not trying to call David Ishii a nerd because he's a physical, athletic person to a degree, just like most people. But he's very technical, and he's smart. Don't get me wrong; he's very smart, and he's actually athletic. He's strong. He did a lot of martial arts and stuff. But when it comes to training, he's not a canine trainer. Okay, and as a result of that, he's going to have a harder time assessing traits. And a dog breeder needs to be able to assess traits, and that's the beginning to really putting the geneticist and breeder combined into one thing. So we all have our own skill set, and it sounds like you can. I hesitate with some of this stuff because it sounds like I'm throwing under the bus. But before I throw him under the bus, remember I said myself, I don't consider myself a canine geneticist. I consider myself more like a master mechanic. He's more on the canine geneticist side, if that makes sense. So we have different skills, and that's probably why he and I work well together, because he understands some stuff that maybe I don't, and I understand some stuff that he doesn't, and so together, because we're cooperative and we respect each other, we're able to um, share our knowledge and both gain as a result. And this is one of the problems in the canine world, is so many people want to pretend like they're an expert at everything, because if they show any shortcomings, they're insecure and feel like they're going to be look bad. They're going to look bad, and that's not good. You should not feel like you're going to look bad just because you asked a question. I can ask you how to make pumpkin pie because I don't know how to make pumpkin pie. That doesn't mean I can't cook a steak. Okay, I just don't know how to make pumpkin pie. But some people are afraid to. If I ask how to make pumpkin pie, it's going to make it look like I can't cook. And if I can't cook, I can't cook a steak. Grow up, get some testicles, and don't be so insecure. All right. Okay, I'm not going to show that. Lee knows what he's talking about. I appreciate that. Dogman, yeah, for sure. I've been up on Lee for a long time. Appreciate it. Trey, basically, you need to draw blood to know. Sometimes you do, sometimes you don't. All right. So for knowing who the parent is, you might need to do a DNA test. For tracking the heritability of a monogenic trait, you don't need a DNA test if you have a good understanding of the dogs and the pedigree, and people are telling the truth. Unfortunately, some people lie, and I can look at a pedigree sometimes and see if somebody's lying, because there are traits that you can identify in a pedigree. For example, if I see two fawn dogs and they had black offspring, I don't buy that. Two black dogs can produce black. Two black dogs can produce fawn, but two fawn dogs can produce fawn. Two fawn dogs cannot produce black. And I saw people argue and say, a guy said that, and they were like calling it, saying he was wrong. Well, if you think he's wrong, then you don't understand genetics. Or you saw a fawn dog bred with a red nose, and it made a black, and you thought it was two fawns, but it wasn't. One of them was red nose. Or if you saw a fawn breed with a chocolate, and you saw the puppy get black, that can happen. So a fawn can breed with a red nose and get black. A fawn can breed with a chocolate and get black. A fawn can breed with a blue and get black. But a fawn bred with a fawn cannot make black. That's just the way genetics works. So next, isn't the pigment that creates the fur color it's the pigment that creates the fur color. Yeah, I do understand it's also genetics. That's right. So yeah, pigment creates fur color and skin color and nose color. All right. And sometimes those colors are the same and sometimes they're not. The hair sometimes matches the skin and sometimes it does not. And then you have brindle, which is another situation where brindle dominates fawn and black dominates brindle. So you have multiple alleles for that loci. All right, scrolling through. Google is fine. All right, good. Y'all could hear me when it is. My computer said my mic went out, or said quit sharing the screen, and then, but it looked like it was still working. Intriguing for sure, above my pay grade. I'm glad I don't breed for color. Exactly. Don't worry about color. There's a few colors that I try to avoid. One of them is white. White is associated with genetic disease because it can be linked with blindness and deafness. I don't like white dogs for that reason. You breed a homozygous white with a homozygous white, you're going to end up with blind or deaf dogs. I also avoid merle because merle is a condition that results from a dominant gene that causes the blocking of pigment when you have a homozygous merle combination. So I don't want merle in my population either because it can lead to blindness and deafness because it can block pigment expression. Pigment expression is necessary for the ear and the eye to develop properly. You need some pigment, whether it be red pigment or black pigment, you need some pigment. If you have an albino, you don't get pigment expression, and then you end up with blind and deaf dogs because the eye and the ear does not develop properly. All right. Yeah, don't try to learn genetics all in one day. It's not going to happen. You just need to take time. That's why I showed you the description in the video. If I go back to that video description, which I'll do real quick, here's how you learn genetics. If you want to learn genetics, do this right here. Do not try to learn it all in one day, y'all. Okay, let's see. Oh, huh. All right. So if you want to learn genetics, sorry about the freezing there, go through this post right. Oop, go through this post right here and look at these concepts one at a time. Learn what haploid is. Learn what diploid is. Skip meiosis for now. You can come back to that a little bit later. But you definitely want to learn meiosis eventually if you want to learn genetics. Learn what monogenic means. Learn what polygenic means. Learn what dominant really means. Don't get it confused with allelic frequency. Learn what recessive means. I should have added to this. All right, let me go ahead and add to it something here real quick. Learn two other concepts. Learn complete, or three other concepts: complete dominance, incomplete dominance, and codominance. Okay, learn those traits or those terms. Just go through this list and learn one thing at a time. Don't bother learning the next one until you understand one. One of the problems with school is we allow people to pass a subject when they have a B, an 80%. Don't do that. Don't go on to the next subject until you have 100% understanding of one subject. When you have 100% understanding of one subject, you move to the next. This is what I taught my son to do. He graduated high school, first in his class. His lowest grade, taking honors in college, honors courses, honors calculus, honors chemistry, and other honors courses. His lowest grade was a 96. Okay. What does he have to do before he takes his next courses? He needs to find those courses that he got a 96 in and go back and study that 4% that he was missing. You need to graduate with perfection. You move to the next level when you've mastered the previous level. You might think that's ridiculous, Lee. Well, what if your mechanic could only fix 96% of your car? Are you going to let them take your car apart? No, they need to know 100% of the car. Okay. Now, I'm signed into on my website, I have a members only section and I have a tab on Canine Genetics. That's where I got this picture from. If you want to read, I've got a bunch of stuff here about genetics. I'm going to scroll fast because this is a pay-per-view page. All right. If you want to learn more about genetics, I have some information about basic concepts that every breeder should know. If you don't want to pay to read these private sections on my website, that's fine. I'm not asking you to join. But if you want to read these, you can sign up by membership. The membership is $100 a year, which is $8 and what, 30 cents or a month, and you can have access to that. And this members only section of our website is going to grow. All right, we're going to add to that. Okay, so but learn it slow. Don't try to learn, and you don't have to join my website. You can research online. The only thing my website does is it puts what I consider to be important information in a very easy to understand method. But it's not easy to understand if you try to learn too much at once. Just do a little bit at a time and ask questions when you get lost. Yeah, I don't care about color or new information to store in my brain for future conversations. I'm taking notes for sure. All right. So you keep females with mostly recessive traits to show if the no, I don't try to keep dogs that have recessive traits. But if the problem, I'm saying you can do what's called a test cross to see if a dog is carrying a dominant trait or not. But it doesn't have to be a female. You can have a male that's recessive traits. Now, that would allow you to breed to a female to see if the female is carrying recessive or dominant traits. Because when you breed to a homozygous recessive, it cannot block anything. And when you can't block anything, that allows you to see if the other parent is heterozygous or if it's homozygous dominant. Because if it's homozygous dominant, all of the offspring are going to be dominant when it's bred to a test cross. If the parent is heterozygous, half the parents or half the offspring will be dominant, half will be recessive if it's bred to a test cross. Now, me personally, I don't want a population of offspring that shows undesirable traits, which means I'm not going to do a test cross if the recessive is undesired. I'm not going to keep an undesired dog in my yard. I got to feed it and take care of it. But sometimes a recessive trait is desired. Just because it's recessive doesn't mean it's weak in performance. It might be a recessive gene like red nose. It's not undesired. It's just a color. It's not linked to any genetic disease. It's just a color. Blue is just a color. I don't want to see it in game bred lines because I think the pedigrees are falsified if you see it in game bred lines. But if it was in the game bred lines, it wouldn't matter because it's just a color. Brown is just a color and it's a recessive modifier. So if the recessive modifier or recessive gene doesn't matter, maybe I'll do that breeding, but I'm not doing it for a test cross. I'm doing it because I like that combination. And then when I do that combination, I'll look at the offspring, and by looking at the offspring, I'll determine if that mother was heterozygous or homozygous dominant, if that makes sense. So you don't do the breeding when you have a small population because you got to feed these dogs. You don't do a breeding to see if they're a carrier of a recessive or if they're homozygous dominant. But if you did the breeding because you wanted to do the breeding, you observe what was the result? Was it 50-50 or was it 100% dominant? If it's 100% dominant, then the unknown individual is homozygous dominant. If it's 50-50, then the unknown individual was heterozygous dominant recessive. Okay, had both. I hope that makes sense. Now, if you had a university that was financing your breeding program and you could have hundreds of dogs and a team of 5 people that were going to help take care of them, then yeah, you could do the breeding just to find out if that unknown individual is homozygous dominant or heterozygous. But most of us don't have a team of 10 people and room for 100 dogs. So in that case, I would not do the breeding to find out. I would do the breeding if I wanted to, and then I'd observe and find out. Okay. So this takes years to complete. It does. It's a lot of work. Mr. Robinson, not Robertson, but I'm sure that's a typo. Robinson. What the guy you have on your YouTube channel last night, what is the name of his YouTube channel? Would love to subscribe to it. Go watch that video and go to the end of the video. At the end of the video, we mention his, you can find him by going to the number 4, and then so THE, then the number 4, and then ORCE multiplier. The force multiplier with take out the F and put in the number 4, the force multiplier. Now he does have the complete, I don't know if it was the complete canine or what, but go watch that video and look at his YouTube name, and I'm sure you can find him. His name is Brandon Edmonds. He's on Facebook. I think he's on Instagram. I'm not sure. I've always gotten along with him just fine. He and I have differences of opinions on various things, but so what? We're respectful to each other. And like I said, with me and David Ishii, we have different opinions, different experiences, and different skill sets, and we're respectful to each other. So as long as people are respectful to each other, you can work with them. But sometimes people poke the bear, and when they poke the bear, they get surprised when they poke back. And Brendan Edmonds does poke a little bit. He does poke the bear a little bit, and then he gets poked back, or maybe they poked him and he pokes back, and it creates a little, he's got a little bit of controversy around his name. But personally, I found him to always be respectful to me. I'm respectful back. We disagree and we discuss things like men, and we were able to move forward. And I find him to be fine, but I do know that he will poke the bear or get poked back, and things can escalate. But, you know, so what? Be respectful to him. I think he'll treat you right. The Structured Canine Academy. Thank you. Understanding this, I'm lost. Look, it's going to happen, especially when you get into the Punnett squares, and I can't design it. I wish I could. I need a page with example problems for me to go through the Punnett squares to show some of this. I do understand that part. This is a master's class. Look, we are going to have a series specifically on teaching canine genetics. So if you're interested in a very specific course of canine genetics, we are going to provide that, and we're going to start off at level 1. And we're not going to progress as fast as we did in this video. We're going to master one thing and then move on to the next and then move on to the next. If you're like, I don't want to do that, cool, don't do that. If you do want to do it, Sign up for it, and we'll try to keep it simple. It's like algebra 3 and 4. No, it's maybe like algebra 2. But yeah, I hear you. And hey, you might be good at math. If you're good at math and you're good at algebra, you can learn this stuff because math and science, they kind of go hand in hand. Are you explaining the buckskin bred to the red-nosed female and all through black and chocolates or on the yellow group? Something along that line, yeah. So people were saying that two fawns cannot produce a black. Well, one of those fawns was not a fawn. It was a red nose, I'm sure, or it was a chocolate. And if you have a red nose or a chocolate, then you can get blacks. Okay. But if it's two true buckskins, you're not going to get black. According to the PED, that female had 1 black in 4 gens. Yeah, I didn't look at the PED. Somebody made a comment saying you can't breed 2 fawns and get blacks, and they said they were wrong. And I was like, no, actually, he's right. All right, but if it's a red dog, that's not a fawn dog, and if it's a chocolate dog, it's not a fawn dog. And people didn't know the difference, I guess. All right. I used Wisdom Panel Premium Dog DNA Test, and it had all the DNA charts on it. I was well impressed. Very much enjoyed this, Lee. Awesome work again. Thank you. The bully dog guys would love this information. Yeah, because they breed for color. And look, I've never really studied coat color that much. I just learned some examples of it because coat color is an easy thing to look at and identify and trace. And so it's a good example for genetics. Just a genetic conversation. Buckskin dog with only one ancestor being black to a red-nosed female, those with all the reds in the PED, no blacks will not produce blacks. That's incorrect. If she's a red-nosed female, that red-nosed female could carry a black gene and then block it because red nose is on a different loci. So when we went back to, if we go back to earlier in the video when I showed you the karyotype. All right, so this is a karyotype. So you have the black and fawn on this location. So you have a fawn dog, which is 2 little B's, and then it was bred to a red dog that may have had a black gene here. But you didn't know it had a black gene because on a different loci, a different chromosome, a different location in the genome, there was a red modifier. And so the recessive red on this turned off the black genes that were present in the red dog. But when that red dog had its babies, the red recessive was blocked by the fawn dog because the fawn dog did not have the recessive modifier. So the fawn dog expressed the color that's shown here, and then the red dog had a black gene here. So you can actually breed a red dog with a fawn and have black puppies. A red with a fawn can produce black, but 2 fawns cannot. Okay, next. Dogman, Aaron, mind blown today, Lee. You really excel at this. Well, I appreciate it. Good evening, Mr. Robinson. It's morning here. I'm not sure where you're from. You can IVF female transplant with dogs, in vitro fertilization, and embryo transplant. Yes, black. Believe it or not, I got a picture of this. I'm going to show it to you. Back when I was in college, I did an embryo transfer, surgical embryo transfer, and it was me. I don't have it on my page. I know I do, but oh well. But when I was in college, we did an embryo transfer. We took a baby sheep out of one sheep and transferred it into another sheep, and the other sheep got pregnant with the first sheep's baby. But it was super small. I mean, it was an embryo. It was just as tiny as a pin dot, right? Early, before it developed body parts. So, you know, it's like just maybe a few days or a week old or something like that. It was probably just actually a few days old because it had to be done. The embryo would have had to been removed before implantation in the uterus. Okay, I've seen many dogs develop like crap, rye jaw, and they finish maturity after 3 years. They may have been old and bred 2 or 3 times, pushing that crap into the gene pool. There's a reason to wait. You need to wait until you know what the dog is showing, and sometimes you can wait less if you know a lot about the pedigree and the ancestors of that dog. But risky anytime you breed early. Theories and calculations, what's supposed to happen on paper versus what happens in real life. A lot of people don't understand the difference between probability and frequency and real life. The difference between them is when you do something many, many, many times, frequency approaches probability. And this is it's on paper, but it's also mathematic truth. So if I flip a quarter one time, the probability is 50-50, but the frequency is 100%. It's 100% and 0%. 100% heads, 0% tails, or 0% tails and 100% heads. And anybody that disagrees with that because of real life experience doesn't understand how accurate theory is. Now, if you increase that test to 1,000 tosses, it's going to approach the frequency of how many times it happened is going to approach the probability. The more you flip it, the greater it becomes closer to 50-50. That's the mathematical truth of how it works. You flip it twice, you could get two heads, you could get two tails, you could get heads, tails, could get tails, heads. So you do it just a few times, like 4 times in that example, you're not necessarily going to get 50-50. You might get heads, 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 tails. But when you do it more often in real life, frequency approaches probability. And that's what people don't understand. They have small, limited sample sizes. And because they have small sample sizes, they say, well, I thought you said I was going to get 50% this and 50% that, but I didn't. I got 2 black dogs and 0 fawn dogs. Well, that's because you only had 2 puppies. Okay, do this 200 times and you'll find out that there's a reason half the world is male and half the world is female. That's because there's 8 billion people on the planet. But if you have, you probably know somebody that had 2 kids and they were the same gender. Because in a small sample of 2, it doesn't always approach, frequency doesn't always approach probability. But in a large sample, it does. What about the dilution relation of alopecia of the dilute blue Isabella dogs? For me, that's a good reason not to bring in dogs with the recessive genes for it. Some people stay away from blue because they say blue is linked to an increased expression of mange or other diseases. I haven't seen that myself. We've had blue dogs and no skin problems whatsoever. But in some populations, it might be. However, if that's the case, you're probably dealing with weaker genes. Dogs that have weaker genetics in other areas for health. Maybe a weaker immune system or autoimmune deficiencies or something like that. That's my opinion. Don't take it for what it's worth. I did not speak, like when I was doing this topic here, I did not speak like I know for a fact. Okay, this is my opinion. So please hear what I say. If I say something is a fact, you can pretty much take it to the bank that it's a fact. If I say I think or it's my opinion, don't take it as a fact. Just take it as an opinion and what I think. But I'm not 100% sure. With American Bulldogs, the rule of thumb has been always red nose to a red nose cannot produce black nose. All right, red nose to red nose will not produce black nose, but red nose to a fawn can. Okay? I've never seen 2 red nose produce a black nose. I agree. It should not happen. And if it did happen, I would question the pedigree. So if you see somebody breeding 2 red nose dogs together and they get puppies that are black, here's what happened. I guarantee you. One of the red noses was not a red nose. It was a chocolate. So because a chocolate is a black modified dog. Okay. So I don't know if you're still here, EBKC, but if you ever see anybody advertise red nose to red nose and they have black nose puppies, that means one of the red nose parents was not a red nose. It was actually a chocolate. And a chocolate dog can look like a red nose. I've seen it. Sometimes chocolate is usually brown, right? But in some cases, it can actually be. Let's look at a dog from Madison Parker's place. So we're going to go to Bulletproof Kennels. I know he's got a chocolate dog, and we're going to share his screen, his website. He has a female named Boots a Blacksmith. And that's her right there. Now, this is a chocolate dog, but some people might, that with limited experience, might think that that's a red nose. That's a chocolate or a liver color. If she was bred to a red nose, she could have black dogs with a black nose. And some people might say, well, that's a red nose. Well, it's not a red nose. If the male was a red nose, let's look at his males, see if he has any. Yeah, my computer froze up again. Oh, there it goes. So let's go back. Look at his studs. See if he has any red nose studs right now. So there's this is Uncle Chunk, I believe. So Uncle Chunk, I believe his mother is Boots of Blacksmith. Yep. So Uncle Chunk is Boots of Blacksmith, and somebody might look at this dog and think that's a red nose dog. It could be confused from that by a quick glance, but this is actually a red dog with a chocolate nose. And so if you take a red dog with a chocolate nose and you bred it with a red nose, you might think all the puppies would be red nose, but that's not going to be what happens because this is not a red nose. This is a red dog with a chocolate nose. And a chocolate nose is a black modified gene. It's not a red nose. All right. Isabella is when the liver chocolate gene further dilutes the blue gene. Interesting. Now, I personally would want to look into that because I'm curious if it's actually a red nose gene that dilutes the blue gene, or if it's a liver. No, you wrote a liver chocolate gene. Okay, good. I would agree with that. So yes, that's a combination of blue and brown dilute at the same time. So I was guessing right. I saw when I searched it, and I thought that's what you were talking about was a tawny or a champagne is what my paths have taught me that a tawny is a chocolate and blue at the same time, and so is a champagne. And I have heard the name Isabella before, but since I don't really get hung up on color, I didn't really, I wasn't 100% sure. And when I searched it, I saw some Isabella Merles, and that's not surprising because Merle does some weird things like that. Have a great day. Have to run. I'll be watching it. Yep, I need to run too. It's been a long one, 2 hours. Okay, thanks for the clarification. And y'all take care. Have a good day. Signing out. Like and subscribe. Support our channel. We appreciate it. We want our channel to grow. Y'all take care and have a blessed day.